All right, today we're zooming way in, down to the molecular level to get a look at one of life's most amazing processes, DNA elongation. This is the story of how life literally copies its own instruction manual, and trust me, it's pretty incredible.
I mean, just stop and think about that for a second. Every single time one of your cells divides, it has to make a perfect, flawless copy of its entire genetic code. This is happening constantly, trillions of times a day. So how on earth does it pull that off with such ridiculous accuracy? Well, the answer is in a process that is just so elegant. The part of this copying process we're focusing on is called elongation. So if the first step is opening up the instruction manual, elongation is the part where you're actually building the copy, brick by brick, or in this case, nucleotide by nucleotide.
But, and this is a big but, this whole building process is not as simple as it sounds. The machinery of life actually faces this huge fundamental challenge that it has to solve every single time. It's what we can call a one-way problem.
Okay, so let's meet our main character in this whole drama, the star enzyme of the show, DNA polymerase. This is the master builder. It's the one that actually adds all the new DNA bases. The thing is, it's a bit of a diva. It has one very strict rule that it will not break.
And here's the rule. It can only, and I mean only, add new nucleotides in one specific direction. Scientists call this the 5' to 3' direction. So you can see the problem, right? It's basically a biological one-way street. Now that's fine for one half of the DNA strand, but what about the other half, the one that runs in the complete opposite direction?
And that is the central puzzle we're looking at. DNA is a two-way street. It's a double helix with these two strands running in opposite directions. But the main building tool, our friend DNA polymerase, can only travel one way. So how in the world does the cell solve this seemingly impossible problem?
Well, this is where nature gets really clever. The cell has come up with this just truly elegant four-step solution to get around this one-way problem. And the best part is, to understand it, all you have to do is remember one simple word. And that one little word is
Plop. Yep, that's it. It might sound a little bit silly, but I promise you this one word holds the key to the entire process of DNA elongation. Let's break it down real quick. The first P is for primer extension, L is for the leading strand, O is for Okazaki Fragments, and that final P stands for proofreading. This is our roadmap. So let's start with the easy part, the smooth side of the street.
Right, so we're going to tackle P and L first. Together, they explain what happens on the easy side of the DNA strand. The side where our one-way builder feels right at home.
Okay, first up, P for primer extension. See, our builder, DNA polymerase, can't just start working on a blank slate. It needs a little starting block, a launch pad to get it going. A short little piece of RNA, called a primer, provides exactly that. Once that primer is down, polymerase can lock on and just get to work.
And that brings us to L, the leading strand. This is the strand that's already pointed in that perfect 5' to 3' direction. So here, DNA polymerase can just cruise. It just follows right behind the unwinding DNA, continuously adding new bases. It's the fast lane of DNA replication. Super smooth, unbroken, and crazy efficient.
So that was the easy side. But what about that other strand? The one running in the, quote, wrong direction. This is where the cell's true genius really comes out to play. This is the O in our plop.
This other strand gets a different name, the lagging strand. Because polymerase can't build on it continuously, it has to get creative. It basically builds it in reverse, in these short, separate little bursts. It synthesizes the strand discontinuously, in small, manageable pieces, moving away from the direction the DNA is unwinding.
And these small pieces have a name. They're called Okazaki Fragments, named after the scientists who discovered them. The best way to think of them is like puzzle pieces. The cell builds one little piece, then it kind of hops back to make another one, and then another, slowly putting together the complete picture.
Of course, you can't just leave a new DNA strand with a bunch of gaps and leftover RNA primers in it. So a cleanup crew of other enzymes comes in. First, those RNA primers that started each fragment get removed. Then, another enzyme called DNA ligase, think of it like molecular glue, comes in and seals all the gaps, stitching those Okazaki fragments together into one perfect continuous strand.
So, we've built both strands, but the job is not quite done yet. Life, as you know, demands near-perfect accuracy, and that brings us to the final and maybe most important P in plop.
You see, DNA polymerase isn't just a builder. It's also a ridiculously meticulous inspector.
It actually proofreads its own work as it goes. So if it accidentally puts in the wrong base, it can stop, back up, literally snip out the mistake, and then put the correct one in its place. This proofreading function is what gives DNA replication its mind-boggling accuracy.
And there you have it. It's a tale of two strands. On one side, you have the leading strand, being built continuously, it's fast and simple. And on the other, you have the lagging strand, built discontinuously, it's a bit slower, pieced together from all those clever Okazaki fragments. Two totally different strategies solving the same problem at the exact same time. It's amazing.
So, the next time you think about how your body heals a cut, or just how you grow, just remember this little mnemonic. From the primer all the way to the proofreading, deep inside your cells, DNA is constantly, and I mean constantly, plopping down new nucleotides to build the code of life.
When you really think about it, it's a process of such staggering complexity and elegance, and it's happening at a scale and a speed that is almost impossible for us to wrap our heads around. It really makes you wonder, when you get down to the fundamental code of life itself,